Hello, and welcome to the Pediatric Brain Tumor Foundation's webinar exploring the use of biomarkers in pediatric brain tumor treatment. Today's webinar is presented in part by Bayer Pharmaceuticals. My name is Heather Held, and I am the National Family Support Programs Manager for the PBTF. We are honored to have you joining us today for this important webinar. The PBTF provides programs and resources to support families facing a pediatric brain tumor diagnosis throughout their journeys. We will provide links to some of those programs and resources at the end of today's webinar. Before we get started, I do have a few guidelines to review. You are muted for this webinar and your video is disabled, but we welcome questions during and after the opening remarks. Please type your questions into the Q&A feature found at the bottom of the Zoom window. We will not get to every question, but we'll try to answer as many as possible. Today, we'll hear from our presenters, and then Kathy Riley, our Vice President of Family Support, will host the Q&A portion of our webinar. I'd like to introduce our first presenter, Valerie Bastani. Ms. Bastani is an oncology nurse with over 20 years of experience. She earned her oncology nursing accreditation in 2004 and has maintained this throughout her career. Valerie serves as an oncology nurse educator on the precision medicine team at Bayer Healthcare Pharmaceuticals. She is responsible for educating clinical staff about adverse event management and oncology disease states. Valerie is passionate about education and patient advocacy and considers working with oncology patients to be a privilege and an invaluable opportunity to learn and grow as both a nurse and a person. It gives me great pleasure to welcome today Valerie Bastani.
Hi, thank you, Heather, and thank you for asking me to be a part of this important webinar today. Today, we're going to talk about the importance of genomic cancer testing and how it can change the way a person's cancer is treated. Now, all of the information that I'm going to present today can be found in more detail on the testyourcancer.com website. So don't worry if you miss anything, you can go to the website and find all of this information. Next slide. So here's a dictionary of terms, and there are many terms that are used when talking about genomic testing. And I know that it can be confusing, especially when different terms are used to describe the same thing, such as genomic testing is also referred to as biomarker testing, tumor profiling, or molecular testing. Different terms, but they are all used to identify genomic alterations. Now, unfortunately, I won't be able to go through all the terms in detail, but I will try to paint a picture using some of these terms to help you better understand some of the words that you may hear when talking with your doctor. It all starts with a gene. Genes are made up of DNA and act as an instruction manual for the body. Sometimes the instructions for DNA malfunction or changes, causing a genomic alteration. This alteration may lead to cancer. So when we talk about genomic alterations, it's important to know that there are different types of alterations, such as acquired mutation, biomarker, and gene mutations, and gene fusions, just to name a few. Genomic cancer testing is used to identify any unique DNA alteration or changes within a cancer cell. When alterations are found, they can tell us how a tumor behaves and what makes it grow. And one of the testing methods used to identify genomic alterations is called next-generation sequencing, or NGS testing. When an actionable genomic alteration is found, it could possibly be matched to an FDA-approved targeted therapy designed to treat that specific alteration, or it could possibly be matched to a clinical study that is investigating a therapy for that specific alteration. And that's what makes a genomic alteration actionable, that there may be a therapy approved or a clinical trial for that person. Another type of testing you may be familiar with is genetic testing. Now, genetic testing is different from genomic testing, where genomic testing looks for alterations in cancer cells Genetic testing looks for inherited traits that are passed on from parent to child, generation after generation. And genetic testing is used to determine if someone has an increased risk of developing cancer due to an inherited trait. Next slide. Science has shown some cancers are driven by genomic alterations. These alterations can cause the growth and spread of cancer. And these cancers can occur in any part of the body. There are no limits on where these cancers can be found. And genomic testing is the only way to find whether genomic alterations exist in a tumor. Next slide. There are several types of genomic alterations which can occur in select solid tumors, such as KRAS, HER2, BRAF, EGFR, ALK, ROS1, and NTRK. Now, NTRK gene fusion is one type of genomic alteration, and this alteration occurs when a normal NTRK gene breaks apart and it fuses with an unrelated gene, creating an NTRK gene fusion. And it's the creation of that fusion that gives the NTRK gene cancer-causing potential. Now, here in the blue are different solid tumors and the frequency in which NTRK gene fusions may be found. In certain pediatric cancers, NTRK gene fusion frequency can be between 10 and 100%. Next slide. We've talked about the importance of genomic testing to identify actionable genomic alterations in general, but when we look at pediatric cancers, approximately 50% of all pediatric cancers have potentially targeted genomic alterations. And when it comes to NTRK gene fusions in pediatric cancers, you can see here different tumor types listed and the frequency of possibility of finding an NTRK gene fusion. For example, infantile fibrosarcoma, 91 to 100% of those babies may be found to have an NTRK gene fusion. Congenital mesoblastic nephroma, 83 to 92% of patients. Papillary thyroid cancer, 26% of patients. And high-grade glioma, 10% of patients. It's so important that every patient with an actionable genomic alteration be identified. Next slide. So why genomic cancer testing matters? It matters because genomic testing is a crucial step in a diagnosis. It gives more precise picture of a patient's individual cancer, and up to 49% of patients who undergo genomic testing are found to have an actionable genomic alteration. So for these patients, there could possibly be an approved therapy or a clinical trial available to them. Currently, more than 100 targeted therapies are approved by the FDA, with many more in development. These numbers, while they represent the percentage of patients who, when tested, have actionable alterations, when you think about the number of targeted therapies now available to patients with cancer, these numbers represent hope. And when you compare where we were as few as 10 years ago with targeted therapies and to what is available today, you can see the great strides that have been made in the development of targeted therapies. This offers new hope to many patients who, in the past, may have had limited therapy options. Next slide. So let's look at the benefits of personalized medicine in cancer care. Personalized medicine, or precision medicine, uses a person's individual genomic makeup to understand their specific cancer, what's driving the tumor, and how best to treat it. Precision medicine helps move treatment beyond a one-size-fits-all approach of traditional medicine, where treatment is based solely on the location of a patient's tumor. So precision medicine treats patients based on their unique genomic makeup. And without genomic testing, you may be missing an important piece of the puzzle when deciding on the best treatment plan for a patient. Next slide. So how to start a conversation with a doctor. I know we sometimes find it hard to start conversations with doctors. We don't always know just quite where to begin. But it's important to remember that you are a partner with your doctor in your care, and it is always okay to advocate for yourself and your child. I find what helps me is writing a list of questions down before I go to see the doctor. It helps keep me focused, and I can be sure that all of my questions are being answered before I leave the office that day. And here are some example questions to help you start a conversation with your doctor about genomic testing. Questions such as, what are the different types of genomic tests? Do you think I should receive a genomic cancer test? And how can the results change our treatment plan? These are all important questions to help you get started with that conversation with your doctor. Next slide. I know that our talk today has been brief, and it's just the beginning of a larger conversation. But I would encourage you to visit the testyourcancer.com website for a complete list of the questions that we just saw, and to learn more about how genomic cancer testing can guide your treatment journey. It's been a pleasure to share this information with you today, and I thank you so much for having me. And Heather, I will turn it back to you.
Thank you so much, Valerie, for laying that foundation for us today as we go into our next speakers. As we continue, I'd like to introduce our next presenter, Dr. Christina Coleman, who will speak to the current state of biomarkers in diagnosing and treating pediatric brain tumors. Christina Coleman is a pediatric neuro-oncologist at UCSF hospitals in Oakland and San Francisco. She is originally from Montreal, Canada, but completed her medical training in the US with her residency in pediatrics at Tufts and her fellowship in pediatric hematology and oncology at UCSF Children's Hospital, Oakland. Her interests include increasing access to clinical trials for all children, molecularly targeted therapies, and long-term follow-up of patients treated for pediatric brain tumors. She is also a mom of 2 children. Dr. Coleman, I'll turn it over to you now.
Thank you so much for having me today. I'm really happy to be here speaking on this important topic and during Brain Tumor Awareness Month also. So we heard some background on what biomarkers are, but today during my talk, I'm going to talk to you a little bit more from the standpoint of a doctor treating kids with brain tumors about how we're using these biomarkers in the clinic today. So next slide. There will be 2 parts to my talk today. I'll start by discussing how we are currently using biomarkers in general terms. And then after that, I will go into some more details and give some examples of how we're currently using biomarkers in the treatment of pediatric brain tumors, focusing on a few different tumor types. Next slide. So in general terms, how do we use biomarkers right now in the clinic? In general terms, I would say we use them to diagnose the tumor type, and then sometimes that can lead us to a certain treatment. I know many of you have been through this, but just as an overview, when a child comes in with a new diagnosis of a brain tumor, we first are getting imaging, and then that often leads to a surgery. That might be a biopsy where the surgeon is just taking a piece of the tumor, or if possible, a resection where the surgeon is attempting to remove the tumor. Once we have a piece of tumor, that tissue then undergoes specialized testing, and a doctor called a pathologist looks at the tissue and does special tests on it to come up with a name for that tumor, which is really important to help us figure out what the best treatment is. And so the pathologist does testing on the tissue, looking at it under the microscope, doing special stains. That's called histology testing. And now what you heard about in the previous talk was this specialized genomic testing that we're doing to further classify the tumor. and sometimes that can lead to targeted therapies. And this can guide treatment, I would say most often right now in the recurrent or relapsed setting, but sometimes upfront for newly diagnosed patients. This information can also be important for clinical trial eligibility. Next slide. So I wanted to talk a little bit about how we name or classify brain tumors. There's something called the World Health Organization classification of tumors, or WHO classification. And this is created by a group of pathologists and clinicians who meet up every few years and review the data that we have based on histology and genomic testing to come up with an integrated molecular diagnosis of tumors. On the left-hand side here, I've listed just one example of some of these names of tumors, and this is just for one subtype of tumor, so pediatric type diffuse high-grade gliomas. And within that category, there are a number of different tumor types based on the genomic findings. The most recent WHO classification came out just last year, and based on new information that we're getting from genomic testing, they identified 22 new tumor types, 13 types that had revised terminology or new names, and then 3 types that used to be subtypes and now are their own category. So this information is constantly changing and being updated as the science advances. Next slide. So you heard a little bit in the last talk about how we identify biomarkers in tumors. So this is specialized testing that we do on the tumor to look at changes in DNA, sort of the instruction manual for the tumor that tells it how to grow and develop, RNA, which is the molecule that follows those instructions to make proteins, and then also epigenetics. So those are changes that can alter which proteins are made. They affect the gene expression to turn genes on and off without actually having changes in the genes. That's all different types of testing that can identify those changes. Right now, I would say we're doing that testing mostly on tumor samples, but there is research being done to identify some of those changes from the tumor within blood or spinal fluid. And I think you'll hear a little bit more about that in the next talk. What's important to know is that historically this testing has really been institution dependent. So what that means is the testing being offered might be different at different hospitals. Some hospitals have their own genomic panels that they offer. For example, at UCSF, we have a test called the UCSF 500 that looks at about 500 different genes known to be involved in tumor development. There's also commercial testing that may be available or different testing at different institutions. This is challenging because then it's not uniform. All kids aren't getting the same testing offered. The good news is that that is changing a little bit now. So if we go to the next slide, I wanted to bring up something called the Molecular Characterization Initiative. And this is an initiative that's been developed between NCI, the National Cancer Institute, and COG, Children's Oncology Group. COG, you might have heard of, is the largest consortium of children's cancer research throughout the country. And they've partnered up to offer comprehensive genomic testing to patients newly recently diagnosed with brain tumors. So any hospital that's part of COG will now have access to this testing for their patients. And this will be offered if families decide to participate in this through Project Every Child. It will be offered free of charge. In about 3 weeks, they will get results of whole exome sequencing, RNA fusion testing, and methylation profiling for the tumors. So this will allow for more equal access to same kind of testing throughout the country for many patients, and will also let us, help us to learn more about the changes we're seeing in tumors. Next slide. So we've heard about how our knowledge is constantly evolving in the classification of brain tumors, how we're naming them more specifically, but how do we apply this knowledge to make a difference in the treatment of patients with brain tumors? So next slide. I'll talk now about a few different tumor types and give some examples of how we're using this knowledge to treat kids. So first of all, in pediatric low-grade glioma, which is the most common type of brain tumor we see in kids, this represents about a third of brain tumors in pediatrics. And overall, this is a heterogeneous group of tumors. So there's some different tumor types within low-grade gliomas. Overall, the survival is excellent, but the morbidity can be significant. That means the side effects from the tumor itself or the treatment can be kind of a chronic illness, really. Traditionally, the mainstay of treatment has been surgery, if possible. And then if more treatment is needed, traditional chemotherapy. So what I mean by traditional or conventional chemotherapy is medicines that usually go through the vein and that target all cells that are growing and dividing quickly. That can cause side effects in normal cells that grow quickly too, like hair or the gut, causing hair loss, nausea, vomiting, or low blood counts. Next slide. In doing genomic testing on low-grade gliomas, we found that there's one main pathway that's activated or changed in this tumor type, and that's the MAP kinase or MAPK pathway. On the right-hand side, I showed a little graph of what that pathway looks like. You can see in the blue are different gene names that can be changed in this tumor type. And in green, they're showing you some different drugs that exist to target some of those changes. So we do have some commercially available drugs that can target some of the changes within this. One of the most common changes we see are in BRAF, something called BRAF fusions, something that's commonly seen. There's been testing that's been done mostly in the recurrent or relapse setting of using MEK inhibitors, something like selumetinib, to target this pathway in pediatric patients with low-grade glioma. So on the left-hand side is a graph from one of the trials where they used a MEK inhibitor for patients who had had previous therapies but had recurrence or growth of their tumor. The 0 line represents the size of the tumor when the kids started the trial, and each bar represents a different patient with a brain tumor. If the bar goes up above the 0 line, that shows that the tumor growed, and if it goes down, it's indicating that the tumor shrunk by a certain amount. So you can see that in this trial, a majority of the patients had shrinkage of their tumor. So this has led to ongoing studies of using these drugs, not only in the recurrent setting, but now in the upfront setting. We're trying to understand if using these targeted inhibitors in low-grade glioma is equivalent or maybe even better than using conventional chemotherapy. And those trials are ongoing now. So that's still a question that's being asked. Next. I wanted to give an example of a patient that we treated with a low-grade glioma. So this is a girl who had a low-grade glioma of her optic pathway, the nerves behind her eyes. You can see in these pictures, we're looking at her brain from up above. Her eyes are in the front. And then that lighter part behind her eyes is the tumor. And when her tumor came back, she had a surgery to try and remove some of it. And then we did specialized genomic testing on the tumor and found that she had a change in BRAF called BRAF V600E mutation. And we have an available drug to target that, which we gave to her. And that led to shrinkage and stability of her tumor. The challenge is that when we stop that drug, it leads to tumor regrowing again. So there are still challenges in understanding the optimal length of treatment, what happens if the tumors develop resistance, and the long-term side effects of these medicines. Next slide. Shifting gears a little bit, I also wanted to talk about high-grade gliomas. When we looked at these tumors specifically, we saw that babies tended to do a little bit better than some of the older kids with these same tumors in general. And a lot of molecular testing has been done on the infant brain tumors and found that they seem to be activated through a pathway called RTK or have alterations in that pathway. So a different pathway than what we talked about in low-grade glioma. And that's characterized by changes in biomarkers. We heard about NTRK, that's one of them, but also all ROS and MET changes. Larotrectinib is a medicine that's FDA approved for tumors with NTRK fusions. And there's ongoing clinical trials now to look at larotrectinib in newly diagnosed high-grade gliomas with these changes. So similarly to what we talked about with the low-grade gliomas, we have some data on using these in recurrent tumors, but still trying to understand their use in newly diagnosed patients. Next slide. Wanted to give an example of a patient we treated. This was a baby born with a high-grade glioma of her spinal cord. You can see that kind of white thickened area in her spinal cord in the first tumor. And she was started on regular chemotherapy. And then when the genomic testing came back showing an NTRK fusion, we started her on the specific targeted inhibitor. And she had a complete response. It doesn't mean every patient would respond that way, but this patient did. And she has no more obvious tumor on her imaging. And she's now a 2-year-old running around and doing great. Next slide. Unfortunately, there are some targets that still remain elusive or challenging. One of those is the tumor category called diffuse midline glioma, or DIPG is another term you might have heard for this. And we've found throughout testing that these tumors are characterized by mutations in something called histones. That's sort of like the spool that DNA is wrapped around. We've had many clinical trials to try and target these changes, but so far we haven't been able to directly target these changes in an effective way. Next slide. I wanted to speak briefly about some of the consortiums that are doing research on pediatric brain tumors. I mentioned Children's Oncology Group, which is doing general pediatric oncology research. One of the groups that does research specifically on brain tumors is called PNOC, or the Pacific Pediatric Neuro-Oncology Consortium, which is run out of UCSF. And this is a group trying to improve outcomes of children and young adults with brain tumors. And they're really focused on pursuing clinical strategies based on the molecular and genetic makeup of tumors. Next slide. I wanted to give an example of a trial that PNOC recently opened that's focused on targeting some of the biomarkers that we found. So this trial is looking specifically at a tumor called medulloblastoma. This is the most common cancerous tumor that we see in patients with pediatric brain tumors. Like I'm showing here on the left-hand slide, this tumor type is roughly divided in 4 different subclasses, and they all behave a little bit differently. We have ongoing studies in newly diagnosed patients looking for to target some of these changes. So we saw that patients with Wnt tumors tend to do really well. So there's a Children's Oncology Group trial trying to give a little less intense therapy to those patients. There are other groups looking at adding sonic hedgehog inhibitors into the sonic hedgehog category. Next slide. In PNOC, we're currently running a trial called PNOC 27, which is looking at targeting the changes found in patients with recurrent or relapsed medulloblastoma. So in the next slide here, I will give just an overview of what this trial is doing. Patients with relapsed medulloblastoma who are a candidate for biopsy or resection would then undergo their surgery. The tumor tissue is then taken for comprehensive genomic analysis and drug screening to try and identify the best treatments for that patient specifically. And then a tumor board meets up to provide these recommendations. And that's just an example of how we're using biomarkers in a current research protocol. Next slide. So in conclusion, I think we've showed that biomarkers are really essential in the diagnosis of pediatric central nervous system tumors. We have some therapies that exist or are being developed to target these changes, but we really need more research to understand the optimal duration and long-term effects of these targeted therapies. I think there's hope that we'll be able to better refine the detection of biomarkers by less invasive measures. So not just brain surgery, but maybe blood or spinal fluid, and use these biomarkers to predict and follow response to treatment. Overall, if we understand the biology, have an effective drug regimen, and can deliver it successfully, I hope that we will make a difference in the outcome of pediatric brain tumors. Thank you.
Thank you, Dr. Coleman, for that in-depth discussion on the use of biomarkers in the clinic. I found the molecular characterization initiative from the NCI especially interesting. I'd like to now introduce our final presenters, Drs. Jack Wadden and Carl Koschmann. Jack Wadden is a postdoctoral researcher in the Koschman Lab at the University of Michigan. Jack is a trained computer engineer, but focuses now on applying his system design and programming skills to the development of rapid, easy to perform, and cost-effective diagnostics for pediatric cancers. Jack's current research focus is on the development of accessible blood-based liquid biopsies to both diagnose pediatric brain tumors and accurately track tumor response to treatment. Dr. Carl Koschmann is a pediatric neuro-oncologist in the Department of Pediatrics and a principal investigator of an independent translational pediatric neuro-oncology laboratory. The Koschman Laboratory is exploring the molecular mechanisms by which recurrent mutations in pediatric high-grade glioma and diffuse midline glioma promote tumor formation and affect treatment response. Dr. Koschmann has designed correlative studies for multiple clinical trials incorporating the use of molecularly targeted therapies for pediatric patients with brain tumors. I now will go ahead and turn it over to Dr. Koschmann.
Thanks, Heather. It's really exciting to be part of this panel today, and appreciate those of you who joined. We're really excited to present some of this research, which is actually directly supported by PBTF and comes from experiments that are being done using patient samples. So we're also really thankful to the families who have contributed to the work that's helping future patients. Moving from Christina's talk, which is sort of where we're at right now, especially in terms of standard of care and pushing the field molecularly, my lab is sort of thinking a few steps ahead and what could the future of care be for children. My work is specifically focused on diffuse midline glioma, but a lot of what we're doing could be relevant to any pediatric brain tumor. Next slide. So just to orient, and Christina talked about this to some extent, but just make sure we're all on the same page. Diffuse midline glioma is a diagnosis that's come about in the last 5 years. The tumor obviously has been around longer than that, but the pathologists now call it diffuse midline glioma. If you have a higher grade glioma in the thalamus, brainstem, or spinal cord that's been biopsied and is in one of those midline structures, and a majority of those tumors carry this mutation H3K27M, which we know is a hallmark mutation for these tumor types, and one that has implications for the prognosis of the tumor and increasingly for treatments and clinical trials. So it's a very important mutation to test for any midline tumor. Next slide. So this is a survival curve, and for those of you who haven't interpreted these before, if you look at the square on the right, each dot as those lines move is a patient who how long they survived, depending on whether they had the histone mutation in the green lines, the light green and dark green lines, or didn't, and that's the gray line. We call that wild type. So these are all midline glioma tumors, but what you see is patients who have a midline tumor that doesn't have the histone mutation. If you move out past 5 years, there's some percentage of the patients that are still surviving, whereas those with the histone mutations, there are less survivors that make it past 2 years. So we know that as a field, we have a lot of room to improve on the therapies that are available. Next slide. One of the problems we have for midline glioma, as well as really, honestly, every brain tumor, is that our patients are managed in the clinic almost exclusively on the results of their MRI. Obviously, clinicians factor in how the patient's doing clinically, but the MRI carries such a heavy weight. And as some families or clinicians who are on this call may have experienced, oftentimes the MRIs carry a significant amount of uncertainty that there will be a finding and it's not so clear whether the finding represents that a tumor is growing or that there's swelling from a previous therapy such as radiation. Good, next, go ahead, advanced. So this is an example of two cases where it looks relatively similar. There's the white splotch is the what we call contrast enhancement on the MRI. But what on the right, that patient went on to have the tumor was growing. We call that true progression. Whereas on the left, that was what we call pseudoprogression. It means the patient had edema or swelling from previous radiation. And so we found this, lesion on MRI, but the patient didn't go on to have the tumor grow. And it's very hard for radiologists and clinicians to be able to see the difference. Next slide. So one of the things that Christina touched on and Jack and I are going to go deeper on is whether we can use liquid samples from patients without needing to do a neurosurgical procedure to be able to understand in real time whether the tumor is responding or not to treatment. And so this is, these are called liquid biopsies. And in the cancer setting in general, a lot of progress has been made in just taking blood samples for patients who have tumors or leukemia that are in the body. But for brain tumors, we know that the DNA that's leaked from tumors, which is what we're searching for in liquid biopsy, is only very infrequently found or in very small amounts in the blood. And so for brain tumors, our group and others have shown that the DNA that leaks from tumors is really enriched in spinal fluid. So that's the fluid that's made in the brain and coats the brain and spinal cord. And it's really a space where if you were able to sample it either through a lumbar puncture or through some sort of ventricular access in the upper brain, we can find the DNA from the tumor. And we're trying to figure out what information we can get when we get those samples. So go ahead and advance. This is one of our first patients that we tried this on. It was a patient with the DIPG whose tumor had the mutation, the H3K27M, and the patient was undergoing a shunt placement where this neurosurgeon was placing a tube in the ventricle to relieve pressure. And so spinal fluid was being thrown away, and we sent that spinal fluid to our lab, and we spent a long time developing an assay where we could assess for DNA, each one of these droplets here, using this machine called a droplet digital PCR. The green droplets are little droplets that are filled with sample from this patient that had only the wild type or the normal histone gene, and the blue droplets are those with the mutant. And so what we do, and a lot of the work you'll see from Jack and I, is it's a ratio of the mutant to the total number of droplets. And that's we call the variant allele fraction. And it's basically the percentage of DNA in that spinal fluid sample or any liquid sample that carries a mutation. Next slide. So we started to feel convinced, increasingly able to have feasibility of finding this in spinal fluid. But for high-grade glioma, it's not part of standard of care to collect spinal fluid really at all. Maybe at baseline, some of our patients receive a lumbar puncture, but otherwise not. And that's different for different types of cancers. So for medulloblastoma, lumbar punctures are done more frequently, or for leukemia, where lumbar punctures are done more regularly. So the pediatric oncology clinics have the infrastructure to collect spinal fluid and have a sense of when it's safe to do it and when not. But because we haven't had a reason to do it clinically, there's nothing we do with the results that affects therapy. It's not a procedure that's done. So we built a protocol for patients on a phase 1 protocol that was being done at multiple sites. And on one arm of this phase 1 trial, we asked families if they would be willing to undergo spinal fluid collection at 3 time points: at baseline, before starting the treatment, and after the second cycle when we did the MRI, and at the 6 months when we did a third MRI. And in all cases, these were patients who were being put to sleep for their MRI. So we did the procedure, but the patient obviously didn't experience it. And we found this to be feasible in about 80 to 85% of the patient visits where we requested it. And we didn't have any issues with safety. We always examined the patient before the MRI to make sure there was no concerns that clinically things were changing that would make the lumbar puncture higher risk. Go ahead, next slide. So this is the result of that, and this is a big, busy slide with a lot of data, and I'll just try to make sure you can understand the high points. All the blue dots are spinal fluid samples from patients with this histone mutation. And the orange dots on the second graph are plasma samples, so from blood samples. And we were able to find the histone mutation in 100% of our spinal fluid samples and in about 90% of our, 90 to 95% of our plasma samples. And what we found, which was actually kind of surprising, is that that VAF actually didn't have any specific relationship, or what we call a correlation, with the size of the tumor. So that was a little disappointing. But then we sort of dug deeper, and maybe you can advance the slide twice, 3 times. Sorry, just one more time. There you go. So this is all the data. This is a lot. But I think the thing that we found that was most interesting was the trend of what happened with the allele fraction over time. So basically, did you have more mutant DNA at the first time point and the last? And that's what we call the change over time. And if it decreased, those patients did better. If they were dropping their mutant allele fraction over time, those patients were going to have a longer response to the drug. Go to the next slide. So this made us interested about, we have this lab value and we have patients' MRIs, and they're not lining up. They're not correlated with each other. So what information can we gather? Is it giving us extra information that the MRI didn't give? And one of the patterns we saw was that for patients whose tumor did grow well on treatment, there was a spike, an increase in the amount of tumor DNA in that liquid sample, often on average about 2 to 3 months before it showed up on MRI. And this is still really a pilot study. So we need a larger study, and we need to do this at multiple institutions to be able to validate this. But in our preliminary data, there was this pattern where the amount of tumor DNA went up in the liquid sample before the tumor growing. Go ahead and advance. So this is one patient who had a brainstem tumor. And if I draw your attention to the plots on the right, just look on the top, that's the orange and gray plot. That gray plot is the size of the tumor over time, right? So you see at 1 month, it was starting to shrink. By 2 months, the tumor appeared to be responding. But after day 120, which is about 4 months, that tumor was definitely growing, not responding to the therapy. And if we draw plasma samples from this patient, we see that that's in orange, that went up in line with the tumor growing, but not necessarily ahead of time. Whereas if you look at the spinal fluid samples, we only had 2 spinal fluid samples, but at that second time point at 2 months, that went up. That allele fraction, that amount of tumor DNA went up before at the same time the tumor appeared to be shrinking, but it was, for this case, early evidence that the tumor was on its way to growing. So we need, again, more cases to be able to confirm that this is consistent across multiple patients and much larger data set. But here this is an example where it gave us some information before the MRI. Go to the next slide. And here's an example of kind of the opposite. So we talked about this issue of pseudoprogression, where the tumor grows on the MRI, the size of the tumor, but we're not sure if it's the tumor really growing or if it's that pseudoprogression where the radiation is causing swelling. So if you look at the plot, you see that gray line that pops up at about 100 days. That was the tumor increasingly getting bigger, but we were only 6 months out from radiation, and we weren't sure that it was the tumor growing versus that pseudoprogression. So we kept the patient on therapy, and sure enough, if you look at her gray tumor, she had a really long response that's still ongoing to this day with this drug ONC201. And if you look at her plasma allele fraction, it only goes down during that same time. And maybe that's a better metric for this tumor, because we know in hindsight that the tumor wasn't actually growing. Go to the next slide. And this is just an example of another situation where we get a medication that can confuse the imaging, but that the plasma samples we got actually helped. It would take me 5 minutes to explain this, so let's just go to the next slide. So I'll just summarize and say that droplet digital PCR is sensitive and specific for these alterations, and that serial spinal fluid collection is feasible and useful, can help predict response to therapy. I didn't get into some of the last dot points there, but Jack is going to move on to sort of the leading edge of what are we working on now. And I'll just summarize to say that our work continues to be research, right? So I want to make sure to clarify that we don't have a test like this that's clinical grade yet, but that we're quite close. And it may come from not necessarily in labs, but maybe from Tempus or Foundation. They're closer to being able to offer tests like this in the clinic, we hope within the next year. Whereas the things in my lab are all through research studies to date. We're not running them just for clinical. So Jack, I'll let you go ahead.
Thanks, Carl. Hi, everyone. I'm Jack Wadden. I'm a postdoctoral fellow in the Koschman Lab. And as Carl mentioned, I kind of work at the kind of cutting edge on the kind of researchy side of improving a lot of our liquid biopsy assays. But with the focus of reducing their cost, improving their speed and accuracy so that we are more easily able to get them CLIA certified and bring them to the clinic. Next slide. So Carl described this really exciting result that we saw in a few patients where, from these liquid samples, both plasma or blood and spinal fluid were able to maybe predict or do a better job at seeing what's going on in the underlying biology of the tumor than MRI. We were using this machine called the digital droplet PCR machine, or ddPCR machine, to take these measurements. But there's a few downsides to this machine that make it difficult to expand to other diseases or even bring this individual test to clinic. One issue is that it's single target tracking. So our assay was validated in looking just at this H3K27M mutation, but of course, as we've been hearing, there are many, many other mutations that might be of interest depending on the particular underlying tumor. And you'd think, oh, just design a bunch of assays for all these different types of mutations, but assay design is very difficult, and there are lots of different mutations that we would like to account for. On top of this machine is actually very expensive. It costs over $100,000, and we are lucky to have one at the University of Michigan, thanks to generous donations from some of these supportive groups. But we would ideally like to design assays that are easier to access, that are available at other institutions or other, less resource-rich institutions than the University of Michigan or institutions like UCSF. So we're using this machine called the ddPCR machine, but what about DNA sequencing? Maybe we can use some of the sequencing technologies mentioned earlier to also track these amounts of tumor DNA. Next slide. And the problem with traditional sequencing is really the turnaround time. So when we submit samples, samples from biopsy, maybe you're getting this full genomic workup, you're identifying, you're sequencing maybe the entire genome of your tumor. This takes for us about 3 to 4 weeks. And I think the fastest turnaround I've seen is about a week, 7 to 8 days. But this is kind of unacceptable in a tracking environment, right? We really want, when the patient is visiting and the family is visiting, we would like answers then, just like with the MRI. So traditional sequencing, again, improves on some of the downsides of ddPCR, but still has these massive deal breakers. On top of that, they're extremely expensive, just like the ddPCR machine. So again, less accessible for kind of a rapid test or diagnostic. Next slide. So instead of traditional sequencing, we started working with this new technology called nanopore sequencing. And nanopore sequencing works by feeding DNA strands through these small little pores embedded in a membrane, and then measuring an electrical disturbance that's created by that DNA across the pore. And you can see that kind of, we call them squiggles on the screen. And by measuring these, feeding these squiggles through a machine learning algorithm, we can recover what actual DNA base pairs were in that piece of DNA and use that to detect certain mutations. And the most exciting part about this technology is that it only costs about $1,000. So you can imagine having, any one of these devices all over the country, even in kind of local, phlebotomy labs. It doesn't even break the top 10 most expensive pieces of lab equipment in our lab, which is ironic, given the power of the device. And it also has, all of the same benefits that traditional NGS sequencing has. We can do multi-target tracking. It's easy to design assays for other targets that a patient might have. And the experiments really can be performed rapidly in lab within about a day. The next slide. So this is the landscape we're looking at. We're taking a lot of the benefits of traditional sequencing, a lot of the benefits of the ddPCR-based assays we've developed, but we're really improving on this accessibility. These are so easy to acquire and use. The one downside is that there's a pretty high error rate, and so that makes it very difficult to detect minute amounts of tumor DNA in samples such as plasma. But we had this hypothesis, hey, maybe we can use this device to track tumor mutations in CSF, where generally there's much more tumor DNA. Next slide. Next slide. Can you guys still hear me?
Yeah, we can still hear you, Jack. I'm not sure.
Okay, great. Yeah. Can you guys see the slides? I can't see any slides anymore.
I think they're coming back.
Oh, I see. Okay, great. All right, great. Thanks, Heather. So I'll keep talking while Heather boots this up, because I know we have limited time and we can kind of see the slide. But Dr. Amy Bruzek had this idea that we could use this nanopore device to measure allele fractions, that tumor content in spinal fluid using this nanopore device. And not only just one target, the H3K27M, but multiple targets. So, the research questions we were asking is this high error rate a deal breaker? Is this going to cause some issues? And the answer is no. The signal stuck out like a sore thumb. So that was really exciting. And also, can we detect multiple mutations in CSF? And the answer is yes. And on this plot on the right, you can see blue is the measure of the size of the tumor, and orange are the signals we're getting from multiple different mutations that were found from this patient's tumor. And you can see that on average, they all kind of track downward with the tumor size, but individual signals are giving these kind of have different shapes and sizes. So this really is motivating multi-target tracking instead of single target tracking to help us better understand and figure out what's going on with the tumor from, combining this with MRIs from these liquid samples. And next slide. So some of my first work coming in was taking Amy's proof of concept work and improving it. The original work was billed as same day. Yes, same day, it's 12 hours, but it's a long day, it's expensive in research time. And so we were able to come up with a new panel and a new workflow that we could accomplish in 4 hours, which was much easier to sell to the hospital as a clinical test. Amy was looking at a limited number of targets. She still had to kind of design these targets for a particular patient, but that's not the easiest thing to do per patient. We would kind of like just a disease-wide panel of some of the more common mutations or gene targets that we see in certain diseases. So for example, DIPG. So our new panel combines a lot of targets into one test. And yes, this was kind of a DIPG focused panel, but we were able to design and validate this test in about a month, which is pretty incredible. And so you can imagine it's very easy to design a similar panel for something like ependymomas or medulloblastomas that we see more in the pediatric space. Next slide. So we got a chance to use this panel in a case study. It was kind of a perfect example of the power of this panel. So we had a patient come in where their tumor was never sequenced. They underwent a biopsy. They were able to recover tissue, but all of that tissue had to be used for histology and traditional diagnostics because that's just the standard of care. They didn't have enough tissue left over from the biopsy in order to send it out for whole genome sequencing. We did know that this patient was positive for this H3K27M mutation via a other standard test. But we decided, hey, let's throw this panel at the patient's spinal fluid to see if not only we can recover and confirm this H3 diagnostic, but also what else can we find? We designed this DIPG panel so we, ideally would pick up some of the more common mutations. And we were able to both confirm that diagnosis as well as find 4 candidate mutations that were not known, and we still don't have sequencing results from this patient. We may never have results, and so it's difficult to confirm that these are real. But this was pretty exciting proof of concept. And on top of that, it only took about 4 hours for not a lot of resources. And next slide. So I know we're running out of time, but some of my new focus now is moving away from spinal fluid testing and really trying to enable this holy grail testing from blood. Spinal fluid is, again, very high signal. It's a really great source of all these biomarkers, but as Carl mentioned, usually we have to put patients under in order to perform that, especially in pediatrics. Children are generally just smaller, and so we get less CSF, and so it's a very, very precious resource in lab when we get it. So it's not ideal in many ways for both patients as well as for research to work with. We would much rather work with plasma. The problem with plasma is that there is such a small amount of the tumor DNA that ends up in patient blood that we really need extreme accuracy in order to resolve that signal in a sea of errors. And this Nanopore device, unfortunately, has a pretty high error rate. Next slide. So a lot of my, again, this is kind of like academic research, but a lot of my work revolves around how to improve the accuracy of the Nanopore sequencer without compromising on time or cost in order to enable these plasma-based diagnostics. And the methodology we're using is using concatemers. So usually when you sequence with the Nanopore, one strand of DNA goes through the pore and you don't get a chance to reread it. You can imagine reading a book, you get lazy, you skim a sentence. I didn't read that correctly. You can go back to the beginning of the sentence and reread to correct your own errors. You can't do that here. So we're designing assays that actually have multiple copies of the original information so that as that DNA is flowing through the pore, we get multiple chances to get it right. And using bioinformatics on the back end, we're able to reduce the false positive rate of this device from about 1.5% down to 0.05%. And that's the great news, because that's the area we need to be in, the accuracy we need to get in order to enable this testing for plasma. So that's it. Next slide. So this is kind of the current, or we're on the threshold of enabling this future where these kind of Nanopore devices really are going to get the best of both worlds for testing. Next slide. Yeah. And I know Carl would like to thank all of our funders, as including me, especially the Pediatric Brain Tumor Foundation for my direct support. So thank you.
Thank you to our presenters. I'm Kathy Riley, Vice President of Family Support for the PBTF, and I'll be monitoring our Q&A today. We've got a few minutes. We'll end at 15 past the top of the hour. Many of you have already submitted questions, so thank you for that. We won't get to every question, but we'll be able to get to some of them today. So I invite Valerie and Doctors Wadden and Koschman to unmute and let us see your faces. First of all, thank you to each one of you. As I was listening to this, it's such a rich, multi-layered discussion. And my first thought from the patient family perspective is I'm so grateful. There are smart people like all of you fighting for our kids. It's more meaningful than I can express. Thank you. Thank you for the excellent work that you're doing, often behind the scenes. For our kids and families. We thank you for that. I want to start briefly. Valerie, you mentioned the advocacy portion of this, and that families are the best advocates to get the testing that's needed. Could you start us off for the families in attendance by crystallizing for us perhaps the one or two important questions that families should be equipped with to ask for genomic testing?
Yeah, I think first thing is just to ask about it. Simply asking the physician, I've heard about genomic testing. Can you tell me where you stand with genomic testing, and how could or if genomic testing can be used for me or my child? I think that's the first place to start. And then learning what that genomic testing can tell and how it may be able to change the treatment plan of a patient if an alteration is found. I think that's the most important thing. But start basic. Just can you tell me about genomic testing and how or does it apply to me?
Thank you for that. We also had a question come in, and Dr. Coleman, I think I'll turn to you for this, and certainly Drs. Koschman and Wadden, please feel free to jump in as well. And this was answered, in this last hour, but I think it's worth talking about. And the question came early on, and it's, do benign tumors still need to have genomic testing?
Sure, that's a great question. And I would say yes. I think when we're talking about brain tumors in kids, the distinction between benign and cancerous, while it's there, it's a little less important because even a benign tumor can grow and cause problems. So it's important for us to understand what's driving that tumor growth, to think about what options we have to target that.
That's great, thank you. And then for Drs. Wadden and Koschman, we had a question come in about someone who has had a commercial liquid biopsy. And maybe you could help us understand, is that something that if a family had a commercial liquid biopsy that could be used as well, or does the sample need to be from your lab?
Yeah, that's a good question. So there's going to be some variation in that answer depending on the situation. Right now, the state of commercial testing is that a lot of the tests are not necessarily based on pediatric brain tumors. They're panels that are based for the majority of cancers, which are adult cancers. And they're just expanding out those panels. And so sometimes we've seen some results where we know that they're missing the alterations of interest. And I think that will come with time, but it's a question to ask your oncologist as you're interpreting those results. Are Did they even test for the really important ones there? What's an interesting question that has come up increasingly is, for a midline tumor where a neurosurgeon may have recommended a biopsy and a family may have been hesitant, could you use results of a liquid biopsy for trial enrollment? And it really depends on the trial. Mostly not yet, but I think the next wave may be more permissive. The main thing to know, and this comes down to whether it's commercial or whether it's in a research lab, is whether that liquid biopsy testing is CLIA certified. And this gets into a whole discussion in and of itself, but it's the most established way to know that any pathologic test, whether that's blood counts or electrolytes or the way that your tumor is sequenced, is done in a reproducible way that's being accredited. And so for a lot of labs, including ours, our work is research and not CLIA certified, and you probably wouldn't want to take the results and make clinical decisions based on them. Whereas actually the commercial testing is more often CLIA certified than not. And so it is a space where we actually hope there'll be partners as we develop new tests in academic settings, that the commercial vendors are the ones that are rolling it out and making them more available and being billed to insurance and things like that. But it's an area where we have this exciting data, and yet it's probably just still out of reach for clinical use right now for the most of our patients, with rare exception. But just like Valerie was saying, it's worth asking your oncologist about. And most oncologists are already answering questions about liquid biopsy in this clinical setting, and we'll have some information to give you.
Great, thank you. Thank you for that. Dr. Wadden, there was a question that came in at registration that I think I'll direct to you, and I think you'll understand, this perspective from the patient side of things. And you addressed this a little bit, but maybe you could talk just a little more about why results, take so long. And of course, from the patient side of things, we're always thinking, does the delay in results mean bad news?
Yeah, I think to answer your second question first, the answer is no, not necessarily. There are a lot of just economic reasons and functional reasons why tests take a long time. For example, sequencing, because those machines are so expensive, usually institutions will buy a few of them and then consolidate them to some sort of testing facility, and then batch samples. So you will run a lot of different samples all at the same time, and oftentimes they have to kind of wait maybe to get a certain number to justify running. And so that, again, that's kind of one of the nice things about this new low-cost sequencer, is we can run whenever we want, on a single patient sample as soon as it arrives to us. In theory, you could do this with one of these machines. It would just be extremely expensive, and wouldn't, I can imagine a university hospital or a hospital wouldn't necessarily, or and insurance wouldn't justify that cost to get that rapid result, which is really, really too bad. So it's a little bit both a technical issue and also, unfortunately, an economic issue as well.
Thank you. Dr. Coleman, we have a question that came in about the NCI-COG molecular characterization initiative, and wondering what type of samples do they take to do the testing? And then if the cancer has recurred, is it important to get a new biopsy, or can they test the initial tumor?
So this testing right now is being done on newly diagnosed patients, and they're using samples from the tumor to do the tumor testing. So not really the liquid biopsies at this point that we've been talking about. They're also doing testing on blood, and that's the genetic testing that was mentioned earlier, to look for underlying changes that might be inherited and predisposed to tumor development. So that's the testing that's being done right now through that. To answer the second part about, should testing be done at recurrence, I think that's something that we're still learning about. Academically, I would say, yes, we definitely want to understand how tumors are changing when they grow back or return, and there can be changes that are different from the initial sample. That's something we're still learning about, and have to weigh the the risks and benefits of going in there for a second surgery. I think that's another reason why, you know, all this research being done on these liquid, less invasive samples is really important too.
And a follow-up to that we received in our registration as well is, can biomarker testing be done on stored tumor tissue? And anyone can feel free to jump in on this one.
Well, maybe let Dr. Koschmann jump in as well, but yes, there are ways that can be done, definitely.
Yeah, there's really not any downsides to using tissue that's not fresh. I think maybe for tissue that might be over 10 years. But honestly, the technology has gotten really good at taking old, even paraffin embedded tissue and being able to extract information from it. I think the bigger issue, I think, is for some of our cases where it's biopsy only, there often is little to no tissue remaining. And so maybe to Christina's point as well, it's something where parents are very overwhelmed, obviously, when they're going through their child's decision to biopsy or during the biopsy case. But it's actually an important time to advocate for trying to save some tissue for potential molecular testing. And oftentimes by the time we get patients referred to us, the tissue's been fully exhausted. And it does limit ability to enroll on trials or do further molecular testing. And it's just a hard thing for parents to know how to advocate for that specifically. So I think the earlier you can open up the conversations that we've been talking about with your own oncologist and neurosurgeon, the better.
Great, thank you for that. We are nearing the end of our time, unfortunately, but I think based on cutting edge material that you've all presented, and I will say from the patient family side of things, this is a huge learning curve for us, and you've broken it down into some nice chunks for us to learn about. But I'd like to go around to each of you, and if you could, in a couple of sentences as we conclude, what most excites you about the future of your work in genomic testing? And feel free to anyone start, but I'd love to hear from each of you as we conclude the webinar today.
I'm happy to start. One of the things that we hinted at was that spinal fluid is really allowing us to learn things about the tumor, and we think that should be a part of routine care for most brain tumors in the coming years. But actually, I would not be surprised in some of the work that Jack's working on right now is if we can actually get better at learning some of this from blood draws, and so that our brain tumor patients can have the same access that the non-brain tumor patients are getting to liquid biopsies without the need for spinal fluid testing. And I just have to think that within years, maybe 5 years, we'll be at a place where we can routinely monitor where the tumor is at just by doing a blood draw, just like we do blood counts. I mean, unfortunately, I know for families who are going through the battle right now, this needs to happen sooner, but I do think that will happen within the decade.
I'll add that, in addition to what we learned through testing and less invasive measures, my hope is that we can really not just improve the survival of patients, but their quality of life too, ideally by offering some of these more targeted therapies. And as we learn about them and their side effects, be able to improve the life of these kids and families going through these diagnoses.
Yeah, and I will follow up with Dr. Coleman said, the targeted therapies that are being developed is such an exciting thing for patients, knowing that where we have been and where we are going, it's just an exciting time for patients. It offers so much hope. And as you said, with the side effects, the targeted therapies, we would hope that the side effects for these kids would be more manageable than what they're experiencing now. So I think there's good hope for the future, and it's an exciting time for targeted therapy.
Yeah, and briefly, I just want to say less expensive and more ubiquitous and accessible testing. So that's something I'm working on, but I think that's extremely important because we kind of exist in a bubble here at these, university hospitals, these very resource-rich institutions. So hopefully we can democratize this testing a little bit.
Yeah, thank you. Thank you, each one of you, for those excellent thoughts. There is, this is a question. Somebody said, would it be possible to submit a question to be answered later? And yes, please. We'll have some contact information for the PBTF. And if your question wasn't answered today and it's pressing and you want to send it to us, we're happy to get it to our presenters. So I think I will thank you. I asked Jen to share our slides again. So unfortunately, we have reached the end of our time together. I want to thank Valerie Bastani from Bayer, Drs. Coleman, Wadden, and Koschmann for their time today, for sharing their rich material and expertise with us. I also want to thank you, our attendees, for your participation and thoughtful questions. And finally, to thank Bayer Pharmaceuticals for their support of Brain Tumor Awareness Month for the PBTF and for today's webinar. We will be sending those who registered a link to the recording of the webinar and be sending you a portion of the slides at the email address you registered with. You'll see on this slide that the Pediatric Brain Tumor Foundation has several resources listed here to help you in your brain tumor journey, including our survivorship resource guidebook, peer-to-peer mentoring program, and of course, the webinar series, which you've all participated in today. And more information can be found at the links you see here. And then our final slide, the Pediatric Brain Tumor Foundation is here to help no matter where you are in your journey. Please reach out to our family support team at the PBTF if there's anything more we can do to help you. Thank you all. This was a great webinar, and we look forward to seeing you at another one of these webinars soon. Thank you.